Hello, everyone. Thank you so very much for your flexibility with recording online today and viewing this. All the deadlines are still the same, so I hope that you're watching this and it's still Friday, so you have some time to study and to kind of integrate all this information. But again, thank you so much for your flexibility. This will be a one-time thing. So the first thing I want to mention is that in this presentation, we are focused on membranes. Our goal here is to better understand the major functions, the major structure, all the components of a membrane that give it its unique nature and give it its semi-permeable nature. So we're going to start off with the list of functions, which begins here with it being a barrier. And by barrier here, we're not trying to say that nothing can get through, but that it is going to restrict the movement of a large number of different possible molecules. The first thing we want to remind ourselves is that the core of a plasma membrane is hydrophobic, and the outer parts of the membrane are where the hydrophilic nature is. So if a molecule is hydrophilic and it encounters those phosphate heads, but then you see the next thing it would encounter would be these lipid tails, these fatty acid tails, it cannot penetrate through, it cannot pass through that plasma membrane. So hydrophilic become blocked, but hydrophobic molecules have a better chance then of making their way through. Now, it's not quite as simple as just saying hydrophilic and hydrophobic molecules. We've got a little laundry list of molecules here that can make their way through more easily, or those that take a little bit of extra help. Those that make their way through more easily would be things like water, which can diffuse in and out based on the concentration of the other components of those cells. So how high is the internal solute concentration to external? Does water need to be put in or put out? The other thing that moves really easily, non-charged molecules. So those can diffuse here pretty freely across. Now, if we have a charged molecule, or if we have a really large molecule here, a macromolecule, that's not going to be the case. And if that happens, then we have to think a little bit more about different transport mechanisms. So in general, transport is just the idea that certain things are going to be able to move through the membrane. Again, it's really easy for them to simply diffuse across, some it's going to be more challenging. But overall, transport has to be geared to manage the amounts of water and ions and nutrients and waste and all the molecules that are moving through. So again, as I mentioned, water may diffuse in freely, but it's only going to reach a certain amount. We're not going to have an excessive amount of water, hopefully on one side. We don't want a shriveled cell. We don't want an exploding cell. We have to very carefully monitor how much different ions go through the cell based on, we have ideas of charges and the needs of the cell. The cell needs to have adequate nutrients that make their way through that cell. It has to have enough waste that are be able to make their way out. All of these functions are really important within cells. The plasma membrane is also essential with the idea of signaling. So signaling here can take on a couple of different meanings, but the core idea is that the cell needs to be able to respond to its environment, and the cell needs to be able to signal to other cells in many situations some type of message or send some type of instruction. So one thing we could look at is hormones. When I think about hormones, I think about all of the major glands of the human body and the different hormones that are produced. We also have phytohormones and other organisms that make hormones too. But in humans, I think about those related to the estrogens and the testosterones, hormones related to growth. There's all these different signals that are being produced in one part of the body and often travel really long distances to reach these cells where they can have a targeted effect. And the reason they can do this is because there are receptors that are built into the surface of these plasma membranes that allow them to respond to that communication, and then for that effect to take hold inside of those cells. Now we have cells that I think about, their entire job is pretty much signaling. So when we think about dendrites and axons within our nervous system, their entire job is to communicate messages, to transmit electrical signals in order for us to have responses. We've got, signals that are being sent, completely without our knowledge, that are autonomous. And we have others that are really driven by what we're trying to do with our body. So right now, you can't see, but, I am moving my lips and I am moving my hand back and forth when I talk because I like to do that. And all of those things are signals that are being sent right now. My recall of these words as I say them, all of these things are related to that process. We also have immune signaling. So the idea for immune signaling is going to be that our body needs to be able to detect whether a cell is self or not self. So are you a part of me or are you something foreign? Now there's going to be identity markers, things on cells that help to identify that. So, okay, yeah, that is, that's one of my, that's a liver cell, or that's a red blood cell. That's something that I can identify as self. Versus, oh, that's a strange marker on that cell surface. That appears to be a bacteria or something, a fungal cell, or something that appears to be foreign, something that appears to be wrong inside of that host. So we have different ways to communicate that. And then of course, immune cells also have to have the ability to respond. The next one here is adhesion. So when I think about adhesion here, I'm looking at how my cell is going to be able to adhere down to a surface. And that in this example is going to provide for things like crawling, which is kind of a modified here version of motility, which is our next idea, and anchoring. Anchoring for cells is often really important. So you may have a cell that wants to be present in a particular position to maximize getting a resource, maximizing light, to maybe it's a microbe that lives inside the human gut and needs to anchor itself down so it doesn't just get flushed away. There's lots of reasons why a cell may need to anchor down to a particular position. And that's usually here, although I'm sorry, this is a bit blurry, that's going to be things like adhesive proteins or different strategies here for adhesion to be able to attach to that surface. The next thing I want to think about is motility. Now, there are three major motility structures that we see in eukaryotic cells, cilia, pseudopods, and flagella. So cilia here are these bristle-like or hair-like structures that are typically short, that often cover entire organisms. We see these a lot in things like different like unicellular aquatic organisms, like certain aquatic protists or protozoans, that we're going to find these types of structures. What happens is they have this wave-like pattern where water is moved over the top of the surface here, and it causes it to kind of move around and circulate. And as it circulates, then that allows for the movement of this cell. It allows for feeding and it allows for water filtering. Very important in many aquatic versions of these organisms. Now, cilia are really, really popular in a lot of these aquatic protists because of the fact that they allow for the movement of water. This might be a little less advantageous if I was in a terrestrial example. The next example after cilia are pseudopodia, or a pseudopod as shown here. So pseudopod, pod like podia, it always refers to foot. So here you have this foot-like extension where you can kind of have this mechanism to drag and to pull. So it reminds me a little bit of what we just saw with anchoring, except instead of just cementing yourself down to a structure, you can drag yourself along. Pseudopodia are often also used in different like amoebozoa for feeding. But here we're focused on the concept here of how it would facilitate motility. So again, kind of a drag and pull. I'm not saying that you can't find these in aquatic environments. You definitely could. But the idea of dragging and pulling on a surface is probably going to be geared to something that has surfaces. So you might want to think about environmentally where you would expect to find that. When I think about pseudopods, I think, again, a lot of different amoebas. And you may want to look into that as an option for a check-in here that's coming in pretty quickly. The last one here are flagella. Here, one flagella is a flagellum. I should say one flagellum is a flagellum, I guess. Singular for flagella is flagellum. There we go. So I will point out that I think sometimes people use pictures to mean more than they do. So here we have a singular flagellum, but it is possible to have multiple flagella. So what you're really focused here on is the length. So cilia, again, small little bristles versus flagella are much more elongated structures. So if I have a flagellum, I'm looking here to see how kind of far it extends back. Flagellum and cilia also have a different pattern by which they move. Cilia have this like wave-like pattern. You can kind of picture them going like back and forth and back and forth. Eukaryotic flagella are similar. They function kind of like a whip. So it's a bit of a similar mechanism, but a little bit less like wave-like. And then if you have a flagellum in a prokaryote, it functions more like, I would say like a propeller. So it has a rotational movement. So flagella can either have whip or rotation. Cilia beat is what we call them, a beating movement or a wave movement. So what I want you to do for this first discussion question, again, you are probably going to be working by yourselves. If you do talk to someone else in the class and you want to work together, you're more than welcome to, but make sure people both contribute. I would not like for someone just to turn in somebody else's answers. That would of course be not something that would be beneficial. But if you are going to work alongside someone, if you're going to call someone up, or if you're meeting with a fellow student for something else and you want to work through, I think that's totally fine. Otherwise, just plan to work on it by yourself. So draw and describe how cilia, pseudopodia, and flagella differ. So redraw those pictures and give a description based on what I said. Also, if people don't listen to the recording, they're going to miss a bunch of what I said here. And for the next question, in what environments may these different structures be more or less advantageous, is going to be a little challenging to answer. So think about where you would want to swim or have water propel fluid past you, compared to if you have a foot that you're going to use to drag and pull. What environments might that connect to? In humans, the only cell that is flagellated, for example, is a sperm. How does the environment that a sperm moves through compare to different cells of a human body? So think about those types of ideas and work on that question. The next thing is the structure of the membrane. So the structure of the membrane here, we remember, is that we have a pair of these phospholipid and hydrophilic heads, because they're going to contact the water of the extracellular and the intracellular environment, and the hydrophobic tails. So the hydrophobic tails are going to kind of fold up alongside each other. This membrane will curl when these structures are forming, and the things that are afraid of water and don't want to contact water are instead going to contact each other to avoid that connection with the water. So we see here again, we have two layers of hydrophilic making the top and the bottom, and this hydrophobic center that we talked about when we talked about how things are going to move through the membrane and why hydrophilics are unable, or at least less likely, to pass through this unassisted. We mentioned again that this functions as a barrier. So because there's water on the outside and cytoplasm on the inside of the cell, we need to be able to, because we have an extracellular and an intracellular environment, we need to get different molecules to pass through that. And again, as we mentioned, that barrier is going to block many of our hydrophilic ions, also those polar molecules, charged ions, large macromolecules, and it's going to permit instead just those non-polar molecules, those hydrophobic molecules, pretty much water, anything like that. So here's another graphic of that, just to show us kind of how certain things may move. So here what we're looking at the left is the ability of something called diffusion. So diffusion here is when you have something like a hydrophobic and non-polar molecule, like a hydrocarbon, that can just freely dissolve and move through the layers of the membrane to pass through to the other side. In contrast, if we have hydrophilic, they're unable to simply just pass through membranes, and they're going to require, again, one of the types of proteins that would allow them to move from one side or the other. So again, ions, polar molecules, macromolecules, things that have a more difficult time passing the membrane, typically will require a protein to assist them in transport. So again, in this example, if you're looking at this and following through, we have the rapid movement of the hydrophobic, and we have the assist here that's needed for the hydrophilic example. So green here for hydrophobic and blue molecules here for hydrophilic. The next thing that we want to briefly mention is the idea of cholesterol. So cholesterol here is a molecule that we've talked about already that is integrated into the membranes of animal cells. Plants don't have cholesterol. They do have other forms of sterols that do have similar functions, but cholesterol as an individual sterol is not found in plant cells. The cholesterol is integrated here between these different areas or different gaps between these phospholipids, and they're shown as these little yellow clusters. So again, each of these little yellow clusters shown here is an example of one of those cholesterols. Now, cholesterol is really important. I do have another slide because I just wanted to be able to emphasize this a little bit better. But the idea that we're going to see here coming up is that we need to be able to regulate how stiff or how fluid a membrane is. And fluidity here will allow for things to more easily pass from one side to the other, will allow some natural bend and movement of that membrane. So let's look at the role of cholesterol here in a little bit more detail. So again, cholesterol is shown as this molecule that is kind of shoved between two different phospholipids, kind of breaking up this and making the membrane either more rigid or making it more fluid. Now, the reason I say or is because it really depends on the condition that this cell is in. So let's say that we have an animal cell, and the animal cell is being subjected to an increase in temperature. Now, we don't have huge variations in our animal cell conditions because we have homeostasis, we regulate our internal temperature, but other cells and other organisms do have to contend with a little bit more temperature variation than we do. So what happens in those organisms that have a little bit more variation is that as temperatures rise, membranes get to be more fluid, they get looser. And you have to be careful because you don't want a membrane to get to be so loose that it begins to sort of fall apart. Now, if we have something like that, what can happen is cholesterol can actually provide a bit of rigidity because it's an extra molecule in there to kind of act like glue or to kind of hold things together. Now, what can happen if we have the opposite is that if we have cold temperatures, is that a membrane, like other substances, will start to kind of get more rigid. So think about water getting cold and becoming ice. When membranes get cold, they get to be more solid and more rigid as well. It's a lot of fats too in general. If you think about even olive oil, if I throw it in the freezer, will eventually become solid. So assuming it's actual good olive oil, I should point that out, and not diluted down with a bunch of other things. But anyway, side note. So if I have the cholesterol, it breaks up the rigidity of the membrane just enough that it will maintain fluidity at these more moderate low temperatures and allow us, to maintain that. So we have cholesterol that's buffering. We also have the presence of unsaturated and saturated fatty acids that we learned about, those two differences already, that also have a similar effect. So if we have more unsaturated tails, that is also going to make for a more fluid membrane. And if we have more saturated tails, that's going to make for more rigid, more tough membrane. So we have these two kind of mechanisms that we can use that modify the fluidity and the rigidity of a membrane. So the presence of unsaturated and saturated fatty acids and the presence of cholesterol both can kind of act to buffer those effects. Now, the next component here is the idea of protein. So we have proteins that are present in the membrane. Some proteins are what are called peripheral proteins, and they only go through one side of the membrane. And some proteins here are transmembrane. So trans here going through the membrane, meaning that they start on one side, the extracellular, and they go all the way into the intracellular here marked by the cytoplasm. So those are the two major ways that proteins can be situated. And proteins are kind of said that they kind of float, they drift, they're sort of just set into that membrane. Proteins can move, they are not stagnant structures. So most of the time when we talk about proteins, we are talking about their ability to transport. That's their kind of classic idea. So here we've got a protein channel. Channels are, of course, for moving something from inside to out or outside to in or inside to out. But again, as I mentioned, proteins can also be really, really important in signaling. So here we've got a bunch of proteins that have, for instance, proteins that have carbohydrates and glycoproteins that are attached. They may act as a way to detect what type of cell this is or to receive certain signals. We're also going to have proteins that act as like receivers for the inputs from other cells. So a little bit more on the transport proteins, just because I think that's a really important function to talk about when we're talking about the membrane, focusing on it being a semi-permeable barrier. So again, transport proteins are what are going to allow us to move those substances that are very difficult for us to move otherwise. So how do we move these polar molecules? How do we move these hydrophilic molecules, excuse me? How do we move these charged ions? How can we move some of these larger molecules as well, like bigger nutrients and things? We need to have a transport protein system. Now, some transport proteins are really simple, like channel proteins that function mostly based on differences with charges. So here we've got these molecules that are able to move, bypass through that positive charge and through to the other side. We have others that are going to require the use of energy to kind of pump different molecules through. We're going to talk more about the specifics of these different types of proteins as we move through. We really want to make sure here that we just understand that things that are difficult to move through membranes are going to require these proteins for assistance. And we want to remember, again, as I've mentioned several times now, what types of molecules are going to have that difficult time as they move through. So this is an example here of an aquaporin. I like this example because it highlights the fact that channel proteins are not just used for molecules that are difficult to move, but they can also be used for things that have to be moved in bulk quantity. So there are instances where you have to be able to move large quantities of water from the one side of the membrane to the other. And this is a protein that facilitates that really fast movement of water. So going back to then some of these structures of our membrane. So I mentioned before here that we have some proteins that have these carbohydrates that are attached to them. We also will have different lipids that have carbohydrates attached to them as well. So here, let's look at our example of a glycoprotein. So glyco is referring to a sugar, right, a carbohydrate. Protein is referring to the fact there's a protein molecule. So a glycoprotein is a carbohydrate attached to a protein here that is present in the membrane. And then here we have our glycolipid, again, glyco for sugar, lipid because we have a lipid molecule. And again, that's of course, our fatty acid. So instead of having a protein that's embedded here in this membrane, we have a lipid that's already part of the membrane that has a carbohydrate attached to it. And again, that's a glycolipid. So why are these important? Again, as we've kind of talked about here, cells have to be able to communicate with each other. We have to be able to recognize cells as being self or as being foreign. We have to have structures that allow us to adhere onto different structures or maybe to connect with other cells. And all these structures play roles in all of those ideas. So bringing together ideas from this lecture and the previous, draw the plasma membrane and describe its major jobs using its major components. So what I would suggest doing is draw a membrane and then add onto it the proteins and add onto it the phospholipids and then give them all numbers and then use that numbered list to define why they're so important. So let's say I number my phosphate heads number 1, my fatty acid tails number 2. And then on the bottom, I'd write, okay, number 1 is for this, and number 2 is for this. Maybe the proteins I embed in the membrane, I'll mark number 3. Maybe I'll mark cholesterol and I'll put number 4. So give me that really nice numbered list. There should be several items on here. I want this to become a really good study resource for you, more than a assignment that I look forward to grading. So I would really suggest doing that. I think that will be really, really helpful. Okay, that is everything that I have for you in this section. Please reach out to me with any questions or concerns. Knowing that we didn't go over this in person, I totally understand that we did not have a chance to go through all of this. I would say that the one area that I would spend a little time really going through again is really making sure that you can replicate this plasma membrane, really make sure that you can define all of the functions of the components. So what are the major functions of the plasma membrane? And again, how do the components of it help to facilitate that? How does the structure of a plasma membrane dictate its function? All right, again, make sure you write these all up or type them all up. If you're doing drawings and it's easier to do them on paper, absolutely feel free to take a picture of that or scan it in, whichever easier, and put that up into the Dropbox. As long as I can read it, I'm more than happy. Okay, thank you again so much for your flexibility. I hope that you all have wonderful weekends. Take care, everybody.